Hello and welcome to Biology Lesson 2.08 over nucleic acids. So we're continuing our series of 4 videos on the polymers of the 4 macromolecules and nucleic acids. By the way, I'm on page 9 and I have crossed out more about nucleotides, letter F, and above that I've written by the nucleic acids. So again, you were missing a page in your note pack for those of you who got this packet in 2020. And we're trying to revise that. If you're watching this video after 2020, you probably have a correct note packet. So these are nucleic acids are the polymers. We just call them nucleic acids, kind of like with proteins, we just call them proteins. Well, we said the monomers are nucleotides. And I'm going to draw out, so let's do 4 colors. Make a little key here. Let's do blue, let's do purple, let's do pink, and let's do green. Those are some of the colors that I feel like show up the best on my recordings. And we said there's a specific way that these nucleotides go together. So let's make blue adenine. Let's make pink thymine. Let's make purple cytosine. And let's make green guanine. And because it's kind of close to pink, I'm going to do the other one in red. Red and pink are kind of close. This guy is going to be uracil. So how do these guys come together to create what we know as the polymers, nucleic acids? The 2 that we know of that we're going to talk about are DNA and RNA. All right, so let's make a chain here. Let's do an adenine. Let's do an adenine. And then we'll do a guanine. And how about a cytosine? So if we have a string of these, and these guys are going to be hooked together along the side, we have what's called complementary base pairing at CG. So an adenine is going to have a thymine. So we've got 2 thymines in a row. And those are going to come in and connect. And a guanine is going to have a cytosine. And a cytosine is going to have a guanine. And these are hooked together along the side. Excuse me. And when we hook together adenine, thymine, cytosine, and guanine, we have DNA. Now that stands for deoxyribonucleic acid. Don't need to know that, but you might be interested. And initially it starts out straight like this, but it doesn't stay straight. What happens is the entire structure comes in and twists. And you end up with, and I'm not going to redo the colors, I'm just going to kind of draw, well, maybe I will. Structure like this. So we would have the, I'll just do circles. Now, and it looks like it flipped, but it's actually just twisted. They're going to be linked across like this. This bond that connects them here and here is a special bond called a hydrogen bond. So nucleotide is hooked to nucleotide through hydrogen bonds. So here's my monomer, 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 monomer. And these all come together to form this polymer that's DNA. Well, so what's the difference between DNA and RNA? First of all, RNA is always a single strand. And second of all, in RNA, uracil replaces thymine. So if I had a strand of, if I took this, half of this DNA, so let's say I've got my initial DNA, and I'm just going to use letters here. So I've got an adenine, I've got an adenine, I've got a guanine, I've got a cytosine. If this is DNA, if I come in and make a copy, shoot, hold on. So if I want to make an RNA copy, instead of having a thymine, I'm going to use uracil, uracil. And then I'm going to have cytosine and guanine would be the same. My red doesn't look very red there. I'm going to recolor that. The other thing that's different between DNA and RNA, so here we have a double helix shape. It's always found in the nucleus. The other thing about RNA is it can be found in the nucleus or in the cytoplasm. Whoops. Remember, the nucleus is the structure in the center of a eukaryotic cell. The cytoplasm is the jelly-like fluid that surrounds the nucleus. So, two polymers. What's our takeaway here? The monomers, the 5 monomers or nucleotides, adenine, thymine, cytosine, guanine, and uracil, make 2 polymers, DNA and RNA. And there's actually different kinds of RNA. We'll talk about that later. In DNA, adenine and thymine bond cytosine and guanine at CG. It twists into a double helix. It's always found in the nucleus. Oh, look at that. I got a little biology class that's appropriate. I've got a fruit fly on my paper. And they're connected with a hydrogen bond across the 2 chains. RNA is going to be single strand. Uracil replaces thymine. It can be found in the nucleus or the cytoplasm. And so it's going to look a little bit more like this. This would be an example of RNA making a copy that we're going to talk about later. That is 2.08, our lessons on, lesson on nucleic acids. Thanks for watching and have a good day.